Diffusion, the international science radio show.
We have a bouncer on the doors of perception.
The good, the bad, the ugly.
It gets pretty exciting.
The myths, the truths.
Toxicology.
Astroseismology.
Magnetism. The dark side.
Genetically engineered potatoes.
Planetoid.
Planetoid.
I love that word.
Hello and welcome to Diffusion. Sit back and relax while we plant the seeds of weird and wonderful science directly into your imagination. I'm Ian Woolf. On this edition, we hear from Alex Kelly on what's happening at the Biofoundry. But first up, here's news of a virus that makes you fat but healthy. Obesity can be infectious. The global epidemic in obesity might actually be a viral epidemic. Adenoviruses cause the common cold, gastroenteritis, conjunctivitis, and cystitis. There are many different types, and they're easy to catch by touching infected surfaces, having people cough or sneeze on you, drinking contaminated water, or eating contaminated food. The Adenovirus 36 is associated with obesity. And the more obese you are, the more likely Adenovirus 36 is found in larger amounts in your body. Studies have identified the protein made by Adenovirus 36 that causes obesity in humans. From about 1980, there's been an epidemic of obesity in both developed and undeveloped countries. It currently affects more than 500 million people. The fat epidemic doesn't correspond to overeating or under-exercising, but to an actual viral epidemic. Obesity is double the rate in adults and triple the rate in children than it was 20 years ago. The body mass index is the ratio of your body weight to your height, and is used in medicine to determine a healthy weight. A body mass index of less than 18.5 is considered underweight. A body mass index over 25 is overweight, with some risk to your health of diabetes, hypertension, and high serum cholesterol. A body mass index over 30 is obese with a very high risk of harm to your health. In animals, there are 5 infectious agents known to cause obesity, but of those, only adenoviruses also infect humans. The study of infectious obesity started in animal studies in 1990. Caged chickens were getting fat when injected with blood from other fat chickens. Monkeys naturally infected with Adenovirus 36 increased their body weight but showed a reduction of serum cholesterol. Rats infected with Adenovirus 36 increased their weight, their insulin sensitivity, and glucose uptake. It was found that animals infected with Adenovirus 36 generally show increase of body weight and inflammation, increased glucose absorption, and decreased secretion of the hormones leptin and cholesterol. They ate no more than lean animals and did the same amount of exercise, but they still got fatter. Humans infected with Adenovirus 36 also showed lower levels of leptin and cholesterol secretion, with increased insulin sensitivity and glucose uptake. Leptin is produced by fat cells, so normally, when fat cells increase and get bigger, there's an increased secretion of leptin. Leptin tells you to eat enough to maintain your body weight against reduction from exercise. Cholesterol is also secreted by fat cells, and the amount secreted is normally higher in obese people, which increases their risk of heart disease. Lower leptin and cholesterol levels in obese people may be a way to diagnose the Adenovirus 36 infection. This unexpected result also means that people who become obese through Adenovirus 36 infection are less hungry and eat less, and less prone to heart disease and diabetes than people who become obese through other pathways. It's a healthier form of obesity. The Adenovirus 36 infection causes an improvement in glucose uptake in skeletal muscle cells independent of insulin. The virus also causes the cells to have an increased sensitivity to insulin. Both of these effects reduce your chance of developing diabetes. And the way the virus does this is suggesting new drugs to treat diabetes. In obesity that isn't associated with the virus, sensitivity to insulin actually goes down, which can cause diabetes. In human cells in Petri dishes, Adenovirus 36 made stem cells become fat cells, and fat cells to fill with fat faster. And it also caused inflammation. In humans, Adenovirus 36 infection can be diagnosed by the presence of the virus in cells and by the presence of antibodies to Adenovirus 36. In 2005, the first study of Adenovirus 36 infection in people In 2005, the first study of Adenovirus 36 infection in adult humans in the United States showed that about 30% of obese people and 11% of lean people had been infected. The study found that people with the virus were 3 times more likely to become obese. Since then, more than 20 other studies around the world have replicated these results. However, 3 studies found no correlation between fat and Adenovirus 36 in people at all. So it's still a controversial subject. A meta-analysis of 10 studies suggested that the infection is more associated with the accumulation of fat under the skin than of body fat. The second meta-analysis included 18 case control studies, including over 2,500 obese subjects and over 3,000 control subjects. This meta-analysis identified an association between Adenovirus 36 infection and a significantly increased risk of obesity development, especially in children. There is work to produce a vaccine to protect against infection with Adenovirus 36. In mice, injections of Adenovirus 36 inactivated by being exposed to ultraviolet light stopped the mice gaining weight and suffering inflammation after being injected with the live virus. Treatment with a mulberry fruit extract that reduces inflammation has been shown to reduce the chances of developing obesity in mice already infected with Adenovirus 36. So there may be a treatment. While Adenovirus 36 may make you fat, it doesn't make you eat more. It gets the new fat by extracting energy more efficiently from your food. And the virus seems to make you less likely to suffer from heart disease and diabetes. Not everyone who gets infected will become obese, and of course, a bad diet and lack of exercise can still cause obesity without viral help. The paper was published in the journal Virus and was titled Adenovirus 36 and Obesity, an Overview. You're listening to Ian Woolf on Diffusion Science Radio. Send emails to science@diffusionradio.com. We're brought to you across Australia on the Community Radio Network and podcast over the internet on www.diffusionradio.com. Alex Kelly is a molecular biologist with the role of lab manager for the community biology lab, the Biofoundry, based at the Australian Innovation Centre in Redfern. I began by asking him, what are the big changes planned for the Biofoundry?
We're looking at a business model change. So previously we relied upon $40 contributions from our student members, and that would get them access to the lab and mentorship and resources, and we do everything we can to empower them. We still want those members to join, but at the moment we're not asking for a membership fee. Instead, we're looking for a third-party sponsor. This will hopefully be either the Australian Government Board of Education, another party within the Australian government, some philanthropic organisations, or finally some equity investors. And so rather than Biofoundry being a not-for-profit that relies upon community donations in order to run, we'll instead have public and private sponsors, which will allow our students to be able to operate and work in the lab without having to worry about the burden of having to pay for themselves. And it will also allow us to actually hire a manager rather than having a volunteer as we've had for the last 3 years, which will further allow us to do more projects and run more things and to reach out to more people.
And so what sort of changes in structure will this need, or what sort of changes in projects?
Well, we're still going to be operating all of the same projects that we were previously working on, but we're going to be offering individual chances for sponsorship in those projects. So someone can come in and sponsor the Open Insulin project or sponsor the CRISPR-Cas9 kit. At the same time, it's going to allow for sponsors to come in and have a unique tech prospecting opportunity. So they can look in and say, oh, there's this new business that's coming in through Biofoundry that I want to invest in, and then they can have direct access to those entrepreneurs and actually provide investment capital to them. In addition, as a company goes through Biofoundry, we will look to obtain a 5 to 10% stock option so that we will be able to ourselves have a vested interest in the success of that company going forward and hopefully feed the dividend from the success of that company back into future startups so that we can continue to self-perpetuate and ensure that we're creating more value for Australia's scientific community.
Can you tell me more about those kits?
Well, the CRISPR-Cas9 kit itself is fallen into what I would call design hell. I'm currently suffering from the earlier decisions that I've made in terms of how I wanted to go about it, which is not the worst thing because in and of itself, it's a extremely valuable project for myself in terms of developing my skills as a scientist, as well as for the lab in terms of actually having demonstration experiments to teach people. So the failures and the tribulations of the kit has itself turned into many opportunities, but it's still not looking like it's going to be going to market anytime soon. On the bright side, the work I've done on the CRISPR kit has led to the creation of some other kits which hopefully will be going to market pretty soon, especially if I can get some money for boxes and advertising and all of that. So a mini prep kit, a DNA mini prep kit for about 30% of the current cost of the one on the market should be ready to go. So I'm hoping to be selling that around about the $30 to $40 mark for 100 reactions.
And what does that do?
That is for purifying plasmid DNA. So purifying specifically circles of DNA from bacteria. And that's just going to be one of many science kits we're working on going forward. So a gel agarose electrophoresis kit for measuring DNA, as well as hopefully some kind of mini centrifuge kit and mini thermocycler kit are ones that we're interested in. But the actual work on the CRISPR-Cas9 kit has indirectly led to the creation of this gel electrophoresis kit and this mini prep kit.
So the CRISPR-Cas9 kit would have allowed people to use CRISPR to do genetic engineering themselves.
Yes, but not in the way that most people understand it. A lot of people think, oh my god, it's going to allow me to change my own genes. No, it absolutely will not. The CRISPR-Cas9 kit is partly amazing because of how specific it is. It can only affect one species, and it can only affect one gene sequence within that species. That is except, of course, for off-target effects, which is a interesting and poorly understood aspect of CRISPR-Cas9. Long story short, CRISPR is targeted by a 20 base pair sequence, and it's thought that up to 3 mismatches in that sequence can still lead to a binding and cutting event. So what my CRISPR-Cas9 kit actually does, it doesn't let you change your own genes, it doesn't let you make a frog gay or whatever. It's not going to do any of that. What it does is it allows you to take a non-glowing bacteria, make a single base pair change, and make it into a glowing bacteria. Now, I've done the first half of the development of the kit, and that involves making a glowing bacteria not glow, and I did that using site-directed mutagenesis, a classic technique that involves killing the actual organism involved. So that's where you can really see the difference between older techniques and CRISPR-Cas9. The organism is going to live through the process, which, if you're doing it to a baby or a chimpanzee, it matters a great deal more than if you're doing it to a little E. coli cell. No one particularly cares if I accidentally kill my E. coli. The site-directed mutagenesis uses the polymerase chain reaction. It's an ex vivo process, so it's all occurring outside of the organism. The CRISPR-Cas9 change is going to be occurring within a living organism, and that living organism is going to undergo a change that leads to it being able to glow. So I've got a bacteria now sitting in my lab. It's been modified such that it has the gene for green fluorescent protein from the jellyfish, but it's start codon, the instruction inside the gene that tells the bacteria to start transcribing and start translating a protein is broken. It's been broken in a single place very, very specifically. Okay, that's good. I've done that. Then we run into the problem. I've gotten my plasmid from America, which contains the code for Cas9. That is itself a patented product. Who owns the patent is currently the source of a fierce debate, and I don't want to fight with either of the people who are currently in that debate. So I want to create enough change in that Cas9, not the gene itself, but the plasmid carrying it, that I could reasonably claim that it is a unique plasmid. I wouldn't be able to claim it's a unique protein. The Cas9 protein, the wild type one, is patented, but for the purposes of selling the plasmid for a kit, I should at least be able to do so. Now we get to the stage where I made a technical boo-boo. I wanted to be able to have 3 different plasmids inside of this cell line. That meant having 3 different origins of replication, so I chose a new host plasmid to put the Cas9 gene into. This host plasmid is called PRS52, and it has what we call an extremely low copy number. So if you think about the number of individual copies of the plasmid that each E. coli cell has, when we're working with some of the ones we work on, they'll have up to 800 copies of that plasmid per individual cell. So when I cut that cell open, I'll be pulling out 800 copies of plasmid. It's a lot easier to find in the slurry of cell mixture. This one that I've chosen has the lowest of any known plasmid that we work with. It has a copy number of 5. So each individual cell has only 5 plasmids, which means that I have to grow up a very, very large quantity of cells to get a workable amount of DNA. Once I've got that, I then have to work with this plasmid and then put it back into cells and work with this extremely low copy number plasmid to try and achieve the desired result that I want. And so that is the wall that I've currently run into. It's led to some modifications to a lot of the processes we do in the lab, which I think has led to a greater value of kit product once we actually get to that stage. So our mini prep process was purifying a large amount of RNA, which wasn't so much of a problem when we had lots and lots of DNA. But when the ratio of DNA dropped down to the stage where the RNA there's almost 100 times the quantity of RNA to DNA, we had to solve that, and we didn't want to do it through the typical way of purchasing an RNase, that is an expensive enzyme that cuts up RNA, because that's expensive. So instead we found a cool little protocol on the internet which used calcium chloride for the same process. And now our mini prep kit is going to also purify out RNA as well as genomic DNA and all the bits of DNA that we don't want at almost no extra cost.
So once you've got all these different kits together and you've finally got the CRISPR kit together, is using the CRISPR kit to make bacteria glow something that a high school biology lab would be able to do with the kit?
It's something that a 6-year-old would be able to do with the kit. It's going to be a 1-tube reaction, and it's really only going to be about 2 hours worth of effort and not require any special skills for doing that. What we will do with the release of the kit, though, is release 3 different tiers of educational resources. So you're going to have your basic kids science kit instructions, and that's just going to be, hey, look, you made a bacteria glow. Good on you, kid. And then the medium kit is going to go into a much deeper explanation of what's actually going on inside the bacteria. And then the 3rd tier of educational resource is going to be going into design principles. And so that's where you actually learn, how do I design a CRISPR-Cas9 that I can modify to affect a gene of my choice? But no resources within the kit will really empower you to do that. You're going to require a CRISPR-Cas9 that is codon optimized for humanity if you want to, which exists, but won't be in our kit. You're going to need to order a G block for coding that CRISPR-Cas9, and then you're going to need to have some form of delivery mechanism. All of these are very, very complex steps requiring lots and lots of individual resources and potentially even, say, adenoviruses if you're looking for that way of delivering it. So the simple fact of the matter is that even with that none of the tools within this kit is going to empower a person to actually genetically modify a human, but they could teach you some of the principles behind doing so.
Well, that's where I think it's interesting. You've got organisations like CubeWriter that use a little tiny Raspberry Pi computer to let high school kids learn about engineering and space technology within the high school curriculum and actually send a project that will run the software in space. I was wondering if we could do synthetic biology kits that high school students could do as part of the curriculum to learn the basics of biotech that might keep their interest into their university years.
I think that's absolutely what we need to be doing, especially here in Australia where our education system is falling by the wayside. I think that less than a focus on genetic modification, which is in itself a bit of an extreme, it is the kind of thing you want to be doing on the cutting edge of technology. We shouldn't really be focusing our university students towards doing that, or our high school students towards doing that, so much as learning the principles of how it's done, and then focusing on nanosynthesis instead. So the creation of high-value products and molecules inside of living cells, using biotechnology to do that aspect of research rather than genetic modification, which itself has many, many ethical concerns and should really be a frontier of synthetic biology. We should be definitely focusing our high school students on how can they create insulin inside of a bacteria, how can they make a polymerase, and how do all of these individual things fit together. And then we'll teach them about genetic modification, but we're going to leave that, I think, to the experts in the field, people more experienced than us or them.
Because if you can get a project that captures kids' imagination and gives them some of the beginning skills, then you can really get passionate scientists developing out of that, going on to university and changing the world.
Exactly. No, exactly.
So what sort of projects is the Biofoundry doing at the moment, outside of the kits?
One of the more expensive things that you need to purchase in order to do electrophoresis is a DNA ladder size standard. And this is basically 10 fragments of DNA, each of them bigger than the next. So 100 base pairs, 200 base pairs, 300 and so on. And you use that as a size standard to compare your own fragments of DNA and determine what size they are. We recently obtained some plasmids from Penn State University in America. These plasmids can be used to create a ladder, so we're currently working to try and turn that into a little kit, and then we'll try and sell that at about 30% of the market price at the moment. So currently that goes for about $100. We can synthesize and then cut up and purify the ladder for about $10 to $20. We'll sell it for about $30, and we'll undercut the market by a large margin.
And what does that let people do once they've got it?
Well, it allows people to measure the size of their DNA. So that itself is an extremely useful process in research and also the development of various tools. It's also useful for hobbyists and anyone who wants to do electrophoresis. It is something which is it's really a workhorse of the lab. It's something which you regularly use in your day-to-day lab work. And if you're a biohacker, startup space, a DIY biology lab, getting access to something like this at an affordable price is extremely challenging. At the moment, there's only a few niche companies that sell it, and very few people are offering a low-cost alternative.
If people want to come and learn skills at the Biofoundry, how does it work?
Just send a message to our Biofoundry Facebook page, and then you come on in and you hang out with us. We're always in on weekday mornings, and sometimes we hang out for weekday afternoons and weekends as well. And we're trying to make sure that we're open and accessible to everyone. And we're hoping that the Australian government sees the value in what we're doing and seeks to invest in us.
And Meow told me last year that you were looking at a badge system for learning skills. Did that happen?
It hasn't happened yet because we need some investment in order to do some larger scale embroidery. If Meow was embroidering each badge individually, he'd never be able to run for the Flux Party. So the badge system is tying into a more comprehensive, holistic approach to science education. And I really want to reframe this properly and actually do it correctly. And so we're working hard with Andrew Gray from Bioacquisitive and a few other groups to try and develop this education system to the point where we're happy with where it is. But the basic principle is to gamify, modularize, and create a hierarchical system. So the way I always like to envision this is just like an Age of Empires technology tree. So you start off with basic skills at the start, and each of those leads to more complex skills, leading to more complex skills, until you eventually reach the current pinnacle or frontier of where the technology is at. And that way someone can identify an area that they're interested in, which they believe that they want to learn, and find a actual path to being able to achieve that, rather than it being some kind of floaty topic down in the future of their education that they're not really sure when they're going to get to. There's no point learning algebra if you don't know why you're learning algebra. It just becomes a, well, miss, why are we putting letters in here? So I feel the same way about gel agarose electrophoresis, or how I feel about 3D printing. Each of these should be a means towards an end rather than a skill learnt in a vacuum.
So if I had a project in mind that I wanted to get the skills for, I could come to the Biofoundry and learn the different skills until I was up to a level where I could do my own projects.
Yeah, well, right now we're trying to figure out the best way to structure and approach this. So when someone comes into us with a problem, we actually uniquely address it. So we'll say, well, you want to get to here. What do you know already, and what do you need to know in order to get to there? And we will work their way through, and they can come in for multiple sessions until they're confident enough to be able to proceed with their own project. And it's those examples and anecdotes that really inform the process of building this technology tree, building this educational paradigm. So that's what we were trying to do.
If you're an organisation that might want to sponsor the Biofoundry, is the Facebook page the only way to contact you?
There's also my email address, there's also my phone number, but I won't provide those on the podcast. But the Facebook page is the easiest way, unfortunately. Zuckerberg controls a monopoly on all of our lives. But you can also find our website at foundry.bio. When it's not being DDoSed, it occasionally goes down and we have to try and pull it back up. It really needs an overhaul now, unfortunately. Please come in and hang out with us. We love enthusiastic people, and our community is growing all the time, and we'll help you find meaning in this world without basic income.
Alex Kelly, thank you very much.
Thank you very much, Ian.
That was Alex Kelly, molecular biologist and lab manager at the Biofoundry at the Australian Innovation Centre. You can find out more at foundry.bio. I produce Diffusion, which is broadcast around Australia to 28 stations on the Community Radio Network. Diffusion is syndicated globally on the National Science Foundation's Science 360 internet radio station and also on astronomy.fm. Subscribe to the podcast on the Diffusion website, www.diffusionradio.com. That's www.diffusionradio.com, and check the website for links, photos and videos about this week's show. The news music was Rhinos Theme by Kevin MacLeod of Incompetech.com. If you enjoyed this show, you can explore more than 950 previous shows archived on diffusionradio.com, where the shows are labelled by keywords so you can focus in on the stories you want to hear. Join my patrons at patreon.com/diffusionradio. Make a donation through paypal.me/Ian Woolf. Subscribe to the Diffusion YouTube channel at youtube.com/c/diffusionradio. I'm Ian Woolf. Join us inside your audio device of choice for more science wondering next week on Diffusion Science Radio.
Science is fun. It helps you to learn, to know, and to appreciate. When you study science, you may go on field trips. You discover the marvelous interrelationships between all living things. You learn to read the history of the Earth as it is written in rocks and fossils. You find out what makes things tick, everything from a molecule to a living organism. In the study of science is found the most useful and satisfying knowledge of man. Knowledge of his physical world, its past, its present, and its future. And in your moments of relaxation, now and in the years to come, you will find the study of science leading you into fascinating pursuits: photography, collecting. Why study science? Study science because you will find in the study of science a richer, more rewarding life.